And I'd like to even expand that discussion a little bit further. You've been thoughtfully vocal about the unfair comparison between NK cell challenges, if you will, and the current advances and advantages. Can you talk us through a little bit about how this bias might be hindering the innovation for NK cells and perhaps what parallels you see with early T cell development?
Yeah, this is always not being the scientist of my company, I will always be cautious when I talk about science. But what I'm going to say is that if we are in the battle of saying, "Oh, my NK cells are better than your T cells or better than your NK cells," whatever, we're not gonna go anywhere. Autologous CAR T has proven that it works in many diseases, but it's still, as I said, limited number of disease and limited number of patients. It comes with its positive and negative aspects, like every single treatment that we create. The slam dunk of the one size fits all magical pill or magical treatment so far doesn't exist. But something that can have an impact on patient, yes. CAR T cells are incredible soldiers, if I can call it that way. When you engineer them, you tell them, go there, identify this, kill it. This is going to have a huge impact. It's going to work. It's fantastic. It's really impressive, actually. NK cells are different caliber. And unfortunately, many people thought that doing the shortcut of taking whatever we have found in T cells technologies and applying this to NK cells and say, hey, look, it's going to work. Yes, it did work, but it's not really fit for purpose. And one of the things that I insist very often when I try to shy away from that comparison between T cell and NK cell is that if you engineer, in my case, we engineer our cells, NK cells with the proper engineering that is specific to them and fit for purpose for NK cells, you're going to get the best of it. If you use an engineering that was designed for T cells, it may work, but are you really using the best approach? Probably not. So today, more of us are really looking at how do we almost design the perfect approach for each immune cells. And the same way we successfully developed technologies with T cells, we can successfully develop it with NK cells. And I will take an anecdote from a conversation with an investor following our ASH meeting 2 years ago when we had our first in vitro proof of concept showcased as an oral presentation at ASH, which I can feel very proud for my team about because it doesn't happen that easily. And when we were showing how much our engineered cells with this unique and proprietary activation that we integrate into our construct were able to kill over and over multiple cancer cells. In the case, it was leukemia cells from AML cell lines, and how they would be able to kill at a ratio that is very, very high. For one engineered cells would kill 10, 20, 30, 40 cancer cells, when many other type of treatments would just kill 4, 5, or 10 of those. So this is in vitro work, but then someone says, "Oh, your NK cells look like T cells. They're very good killers, like T cells are." And I said, "They don't look like T cells. They are good killers because we engineer them to be good killers. But there are no T cells. They are NK cells, and you shouldn't compare them. You should just look at what they do for what they do." And to close on this statement, I would say the ideal treatment will probably be a combination of T cells, NK cells, macrophage cells, because at the end of the day, they all have something to do to defend the patient from the cancer cells.
Yeah, sure. That makes perfect sense. And to continue the discussion a little bit with T cells, Tracy, with you and your team at NKore, why would you say it's critical to design NK specific activation strategies rather than borrowing activation approaches developed for T cells?
I mean, it's really critical to design NK specific activation strategies because NK cells are fundamentally different from T cells. T cell activation is antigen dependent and relies on engineered receptors, whereas NK cells are innate immune cells governed by a complex balance of activating and inhibitory receptors. you borrow T cell strategies and simply bolt them on to NKs, you risk either exhausting them quickly or failing to unlock their cytotoxic potential, essentially. And by contrast, NK specific activation, when done well, creates a cell that can persist longer, heal more efficiently, and retain its natural safety advantages. NKs require a tailored playbook, not a recycled one, if we want to see them succeed clinically. And one of the more exciting things that we're seeing with our NK cells currently is that actual persistence that we've been lacking in the field and that lack of toxicity. So using very specific NK cell strategies that can really truly unharness their power to not only persist but kill is fundamentally crucial when analyzing and developing these cell strategies.
Gotcha. And Raphael, I'd like to hear from you too. You and your team at NKILT, why is it critical to design NK-specific activation strategies versus borrowing them?
Yeah, so this is actually the core of our technology is based on, like I said, a proprietary fit-for-purpose activation approach. We had a very big study that was made in our lab, and that's what led to actually our oral presentation 2 years ago. But where the idea was to try to tackle two main things that people were saying are no good in NK cells. And I will say, I may not agree with that statement, but I'm no one to say whether it's right or wrong. People were saying NK cells are not good killers or not as good as T cells, and they would say they don't last long enough. They die out after 2 weeks, which is true. Natural killer cells have a kind of a lifespan of a couple of weeks, and then they kind of tank and don't do really anything anymore. We created a very specifically fit-for-purpose design in our construct that we are engineering with our NK cells that is allowing our cells to feed themselves on what helps them maintaining this activation. And what is important is that once they identify the cancer cells, they first attack and kill the cancer cell. But then this process triggers the activation of our cells so that they continue to chase cancer cells and become almost like truly serial killers that will just chase other cancer cells and then kill them. In addition, the specificity of NKILT technology, which makes it unique and very different to many others, this activation triggers a feeding process that allow our cells to remain active not 2 weeks, but hopefully we're going to prove that rapidly in vivo, up to 6 weeks. We already have some evidence that we can reach the 6-week marks and maybe further down the road longer. But our cells remain serial killers, actively killing and chasing cancer cells for up to 6 weeks. And that makes a big difference. Just think about an NK cells that is going to kill for 2 weeks, and suddenly that same cell can kill for 6 weeks. You're just significantly impacting the potential outcome by having a much more and more impactful dent into the tumor. But you also can then have a real impact on how you're going to design the dose and the schedule of your treatment, because it means that you may not need to put so many cells up front, because if your cells are good killers, you need less of them. And so that has an impact on the potential safety outcome also of your treatment, which we know is critical. And NK cells have already a very good safety profile. But if you don't need to put so many of them up front because your cells are going to continuously kill for several weeks, that is also a potential advantage that we want to emphasize, and that hopefully we'll be able to prove in the clinic.